Okay, good morning everybody. Welcome to day 2 of our live online training on metabolomics data analysis in the cloud. My name is James Bradbury and I'm from the University of Birmingham and I work as part of the outreach team for Phenomenal. So our presentation today is called LCMS data analysis with XCMS and MetFrag, and this will be done by Stefan Neumann from IPB Halle. So with that, I will pass over to you, Stefan. Thank you.
Okay, thanks James for the introduction and welcome everybody. So I'm going to talk about LCMS data analysis and we'll show some XCMS and MetFrag, and all of that will be done on Phenomenal. My name is Stefan Neumann. I'm from the Leibniz Institute of Plant Biochemistry in Halle in Germany. And if you wondered where that is, so we here have the map of Germany, and Halle is pretty much in the center of Germany right here, Berlin over there, Hamburg in the north, and Munich in the south. So our institute is part of the Leibniz Association. Our institute itself has about 100 researchers doing plant research, and depending on how you count, there's 5 to 15 bioinformaticians. And we have more than 50 years of metabolomics expertise running LCMS metabolomics for quite a long while. In this session, we are going to do some computational mass spectrometry. And if you have some LCMS data, we first will do a feature detection to see which features are in the raw data. And then we'll do some metabolite annotation with MetFrag so that we get some idea of the metabolite structure of these features we found earlier. I'll also talk about how to use workflows to actually automate such data analysis. And then we'll give a demo of one of the example workflows with example data that we have as part of the every Phenomenal installation. I also give some words about cloud computing in general, what it means for you as users and how to get there in case you haven't done any cloud computing yet, and finish up with a summary. Okay, so when you do metabolite profiling for quite some while, there are a lot of developments. And you see, if you look at the history, that people have done things manually, and they were comparing mass spectra from different samples and then annotating some of the peaks and interpreting their experiment. That's a rather tedious and manual way. And you could imagine that you want to have something more automated. So there's feature detection quantification using different tools out there. Metaline was one of the first, and XCMS is the one I'm going to talk about here, which, if you look at the green stuff, that is the raw data. And then the task of the feature detection is to create the red boxes, which are detected features, and then you can use that for the quantification so that afterwards you can use that in the statistic. And that becomes very, very important if you're having a very large experiment. So here in the upper right, you see the Phenom Center at Imperial College in London in the UK, and they are running 10 mass spectrometers. So you can't even see all of them on this picture. And they are ramping up towards 24,000 MS runs per year, which is a whopping number. And you can clearly imagine that the manual interpretation we did a decade or two ago doesn't fit that scenario anymore. So we are using automated feature detection. We get the data matrix that you can then use for any downstream statistical analysis. And we'll also have to talk by Etienne later in this online webinar series. The second topic in untargeted metabolomics is that you need to identify your metabolites. So in the old days, there was tables like that, which would tell you that if you see a 59 peak, it could be this one, or if you see, let's see, a 79, then it might be that one, and so on. So it was a guide to humans how to interpret the peaks that you actually see in a mass spectrum. And again, that you would want to have in an automated way. So Here we have Mr. Lederberg in the United States at Stanford University quite a long time ago, as you see from the computer equipment in the background. And they created Dendral as one means of interpreting fragment information in MSMS spectra. And later there was like Mass Frontier, which you can still buy from HiChem and Thermo, which helps you interpreting mass spectral data. What we then did here at the IPB, we wanted to not just interpret a single spectrum in the context of a single structure, but we want to have an explanation and scoring of hundreds and thousands of candidates, because you initially don't know which metabolite structure is the most likely behind a spectrum. And for that, we have created MetFrag. MetFrag will go to any molecular database like PubChem or ChemSpider or Keck, whatever, lipid maps, and it will retrieve candidates, candidate structures, and then it will interpret these candidate structures in the context of the experimental spectrum and create some scoring. So we have created a web application where you can specify which database to use, what would be your parent ion. Here, that's the neutral mass of the parent ion to retrieve the candidates. In this case, from Keck, we would see 15 candidates for this particular parent mass. And then you have the MSMS peak list, which is your spectrum. And then MetFrag goes away and crunches through the candidates and will present with you the returned list of ranked candidates. And that certainly gives you an idea of the structure. So fairly certainly this will be some kind of flavonoid. And in fact, it was the rhinanine, which might be here on the first rank that you can see on the screenshot. You can also try to see what substructures were annotated to which of the peaks. Now, the caveat here is that we are only highlighting substructures of the parent molecule and the corresponding mass in the spectrum. We are not claiming that this would be a physical simulation of what's happening in gas phase chemistry inside the tandem mass spectrometry. For that, our approach is not sophisticated enough, but on the other hand, it's much quicker than any energy calculations that you could do in the same time. So the individual tools we have seen are great. In practice in the lab, when you analyze the data from your workflow, from your experiment, you will have some sort of daily workflow. And what happens very often is that you have lots of file conversion and you cut and paste your spectrum into MetFrag and so on, which is pretty tedious. And I didn't even mention all the other tools that you could use as part of your daily workflow. Maybe you would using MS-Dial, maybe you would using mzML and so on. The other thing, if you use some of these graphical interfaces, is keeping track of the intermediate results and the parameters that you have used, because you will need to report that afterwards in the manuscript when you try to publish the experiment. And these issues become worse the more different tools you're using to really get a concise, reproducible summary of what you have done. So rescue comes in the form of workflows. Instead of using monolithic software systems, workflows consist of many connected compounds. And here's an example, rather oldish one on the Taverno system. I'm not sure that the visualization looks the same nowadays anymore. There's some CDK cheminformatics being used here, and you see how the data is flowing from box to box to produce the final output down here. So these execution steps are carried out by those specialized modules. They can be in different languages like Java, Python, R. They could even be on specialized hardware. Currently, I'm not aware of anything, but you could imagine some graphics card accelerated whatever processing software, and that would dramatically speed up on specialized hardware. The execution steps down here, they will wait until the data has arrived from above. So until the preconditions are met, and it can then continue with all its inputs. If there are parallel executions, they can run on different nodes in your compute cluster, and I'm coming back to that later. And another nice thing is that these workflow systems usually support a visual programming style that is much more pleasing and more intuitive to navigate if you're not a hardcore bioinformatician. So in phenomenal and increasingly in metabolomics, people are looking at the Galaxy project as one of the ways to execute workflows. So workflows are put together, again, connecting individual boxes into workflows. There's different visualizations for the data sets. And for every step that you do, there's the data kept in the history of Galaxy, which is a reproducible audit trail of what you have done. You can even rerun a tool using one of those icons. And this is used not only in metabolomics, it's coming from the sequence analysis area, and proteomics is using a lot of Galaxy. So it's pretty mainstream at now. You can see more on the Galaxy websites. There is also a dedicated installation for metabolomics from the French workflow for metabolomics team. And also the team from Birmingham has created a flavor of Galaxy that can do metabolomics. And in phenomenal, so one of the issues is that it's sometimes a bit tedious to set up Galaxy, and you will need the support of your local administration, maybe your local bioinformaticians to set up Galaxy. You see there's a lot of tools involved in there, and setting that up could be fairly tedious. But in Phenomena, what we have done, and Pablo mentioned that yesterday, we are encapsulating the individual tools as Docker containers. And then we have this zoo of Docker containers, which are managed by Kubernetes. And then you can deploy that on a cloud infrastructure. And supported infrastructures could be the Google Cloud, the Amazon Web Services, Microsoft Azure is technically supported. And OpenStack is very important because also many local universities have their own OpenStack cluster if they offer their own cloud installation. So my demo that I'm going to do is the feature detection on the metabolite profiling data with XCMS, annotation of the individual peaks with CAMERA, because one of the peculiarities you see in LCMS is that one metabolite may give rise to more than one feature. So usually you might have the protonated ion flying around, but there could also be the sodium adduct or some insource fragmentation, and all of these belong to the same metabolite. So we would like to sort of use a text marker to say they are all belonging together, and that's what CAMERA is doing. Then we need to extract the MSMS spectra using a different R package and map the MS1 and the MS2 data together. And then we do a batch processing with MetFrag and finally visualize the results. So this is the workflow that we're going to use. It's been created by Christoph Rupkis here at the IPB and Payam Imami from Uppsala in Sweden. And they implemented that in Galaxy, and it's now part of all the installations in Phenomena. The demo I'm going to show, and Pablo mentioned yesterday the deployment, the demo I'm showing is running on the OpenStack instance at the Center for Biotechnology at the University of Bielefeld. And this is part of the scientific cloud operated by the German National Infrastructure of Bioinformatics, which has different locations across Germany. So I'm running now in the Bielefeld part of the cloud. And together, all these cloud sites, they have more than 10,000 CPU cores. more than 100 terabytes, and that's not disk space. This is RAM memory of all these nodes, and a total of 5 petabytes of storage. This has been mentioned how the deployment works by Pablo yesterday, and I'm now proceeding to the online demo. Let's see. This is the Galaxy. And I'm now about to log in. I hope you can still see everything here. No, I don't want to remember that password. And this is now the welcome screen we see on Galaxy. There is some menus up here. And I will start to show you the interactive tours we have in Galaxy. They're a bit hidden. You have to know where to look. It's in the help menu. It's interactive tours. And there are a number of them. Some are created by the whole Galaxy community, covering aspects of the Galaxy user interface, like, for example, that one here. But I'm going to skip that and directly go to the MetFrag workflow. And these Galaxy tours, they are like an overlay of over your Galaxy. They are presenting us with little windows telling us what's going to happen and what to do. And very often you can click next, and it will then even execute the steps to do. So the first thing we have to do is to get our data into Galaxy, and we are using the upload button here. I can click that also by saying next. And this will now open the data import window, and we will download the data from the web. So we fetch the data, that is the URL that we have inserted here, and that will be then downloaded by Galaxy. You click the start button down there, and then it's proceeding to get that into the history. And I'm ending the tour right here. There's a few more steps. In fact, the whole demo that I'm going to give is part of the tour, but I'm executing the steps manually. So to not confuse you with the buttons that we see in the tour and the buttons that are in Galaxy. So I'm ending that right here. And while we wait for the upload to finish, and that one, the history item here on the right turning green, we can also have a look at the other ways how to get the data into Galaxy. So one of them is that we can choose a local file coming from the computer. Because of the screen sharing, you didn't see there was now a browser popping up that I could navigate my hard disk and select a particular file. There's also for larger file sets a way to use FTP to make a connection into the Galaxy instance that we have right here and upload larger numbers of data. We have also seen how to paste data or fetch data from the web if you know the URL. And while we still wait, which takes a total of about 2 minutes for the data to be uploaded, I can also show you that here on the left, there's the toolbox. And the toolbox, there you can search, for example, by keywords. And if we look at the metabolites database, what we get there, then we have one tool that would be downloading the data from metabolites, but we are not right now doing that in this demo. That was exactly on time, fantastic. Now we have this LCMS dataset here on the right. And the first thing that we need to do is to do the feature detection using XCMS. So I can type XCMS, and I will find the XCMS find peak tools right here. I click on that, and these are the parameters that need to be passed to the XCMS tool. Most important, which input file to use. And that is exactly the one here on the right. So you can think of this history here to the right as a directory on your hard disk, so to speak, only that it's living inside the Galaxy. And then we can select items from the history to be processed. There are some parameters on how XCMS is processing that, and we're keeping them at their defaults for this particular data file, and this is now continuing. You see there is a great history item because that is the item we are going to have once the tool has finished. It will now in the background schedule that on the Kubernetes cluster, execute the Docker container that has XCMS inside, and we already see that this has now finished. Then we want to do the annotation of ions in there, and for that we need to use the CAMERA annotate peaks, which is the first one right here. And again, the input is the XCMS find peaks data here on the right, and we can also use the default parameters for this and just execute, get another history item over here. On this history item, we will then do the second CAMERA step, which is the group by the retention time. So all the peaks coming at the very same retention time, they might be coming from the same metabolite. So we're using that. We can already use the annotate peaks data here on the right-hand side as input and execute the job. And this will now do the next step. And the third part we need to do in CAMERA is to run the find adduct step. Again, it's using the input here from the history. I can even execute that before it is finished. Apparently, I just executed that twice. That's not an issue. We now have two, so to say, copies, two results of that. Following up, I will use the one here on the right, the number 6 for the downstream processing. And the next step we have to do is to take care of the MSMS data. So that will be done with the MSnbase. So we are reading the input, again, selecting the output file, which is the one down here, the number 1. So we can execute that, and it's already part of the history. Okay, we now will have the MSMS data extracted. And now we need to map between the MS1 features that were detected by XCMS and connect that to the MSMS data that we extracted here in the last step. And for that, we are using the mapping tool, which is the second here in the list. And now we have some more inputs. First, we need the MSMS data. And second, we need the MS1 information. And that's the output of the CAMERA tool. So once I have selected the correct inputs, I can execute that. And maybe we can take a break and have a look. We can extract the workflow that we have created so far and have a look at that. So this is part of the way Galaxy works. And I can now fire up the editor for this workflow. And this is sort of half the workflow that I'm going to show. So there is some input data. XCMS find peaks has been executed. CAMERA has been executed with the first step and the second step. What we are missing is the MSMS data analysis. And we'll see if that is available once I go back to my Analyze Data tab. And this way, we are gradually creating the workflow. We could also drag tools here on the workflow editor, but these are two different approaches to creating your workflow. The one we see here is using the editor and really assembling the workflow. And the other one, yes, I want to leave. And the other one that we have done so far is sort of interactively running the individual tools on particular inputs and then en passant creating the workflow. Okay, we are still waiting for the MSMS data, but we can already continue some steps. The next thing is that we need to create the MetFrag parameters. So in the tools, I'm checking out the MSMS to MetFrag tool. And this will now take the inputs we have here on the right and prepare the queries we are going to run against MetFrag. The one thing is that I need to have the MSMS data that's correctly selected already here. And then we need the camera find adducts, which I find down here. The remaining parameters I can leave. I can also select different upstream databases, but I'll try to do that in CAC because it'll complete faster. You also have the choice of providing your own custom compound database to MetFrag. That would then be provided as a CSV file, but I'm not going to do that. So executing this step now. And this will create a list of datasets. This will now create the parameters for running MetFrag for all the features that have a mapped MSMS data in here. So that's why it calls this a list of datasets. So once these parameters have been created, we will next need to actually execute MetFrag, which we find down here. So we need the LCMS file. Sorry, we don't need that. No, that's why I need to select to the dataset collection. And now you see that there is no collection available because it's not yet finished. And the reason is that this step down here is blocking. And I think I remember that the reason it takes quite a while is because it's downloading the container with the tool still from our repository. And that can take a while because it is a few gigabytes in size. So if this doesn't turn green in the next seconds, I could go back to the presentation and we'll end up finish up the online demo in a minute or two. Okay, so coming back to the presentation, this was the example workflow we have for XCMS, CAMERA, and MetFrag. But there is more. There is, for example, other tools and tool collections out there. A prominent one is OpenMS, which is been created by a larger consortium headed by Oliver Kohlbacher in Tübingen in Germany. OpenMS supports several workflow systems, so it can be run not only inside Galaxy, but it could also run that in KNIME, which is another popular workflow system. The OpenMS tools are called TOP, which are the OpenMS proteomics pipeline tools. And they have been included in Phenomenal also through the OpenMS Docker container. And then you need to configure that into Galaxy. And these steps have been done also by Christoph Rutkies and Payam Imami I mentioned before. This allows to mix and match, for example, OpenMS and the R or XCMS world. So you see here the top tools. There's a large number, many of them dedicated to proteomics, but many of them also applicable to metabolomics. And they are about identification, quantification, visualization, and statistics, and so on. And Payam and Christoph have created this really large workflow where I'm not running through the individual steps. And I'm really only highlighting that right here at the beginning, there is some OpenMS tools that are being run. Then there is some XCMS tools being done. And then you can also convert that back to the OpenMS world. That is a pretty cool feature because it allows you to bring together the tools that otherwise would require a lot of effort to get together. Okay, let's move over back to the Galaxy instance and see how far that has proceeded in the meantime. And we see something I didn't want to see. The red item means that this has failed. And I can now see what was the failure here. And I can also try to rerun that. And maybe I got some of the parameters wrong. Let's see if yes, of course, reading helps. The first input is the camera find adducts. So I select the correct one up here. And the second one is the MSMS data. And I guess most of you will have wondered why I have chosen it in the first time the way I did. And that was because I'm sort of doing many things at the same time right now, including talking, clicking, and everything. And that's how I mixed that up. But that also showed you how to rescue a failing job here. And I can now execute that and hopefully got everything right now. And it will now hopefully create the same, here we go, the same history item, and that'll hopefully turn green very soon. We also see that we are still waiting for the MetFrag parameters, but that requires that step up here to finish. And we'll see if the tool will then resume operation once all the required inputs are done. You also see that the history down here is sort of linear, starting from 1 to 10 now, where the actual workflow that we have already seen before is more a graph structure, where the data is traveling also through different branches. Okay, so with that, we can now run the MetFrag Batch tool. That would be the one down here. I need to switch to the dataset collection that's still not around. So it's still creating the query files in the background. So at the moment, I can't proceed here anymore. And again, if we don't get this finished in the next seconds, I would proceed with the presentation and then come back to that after the presentation. Okay, so we saw we can mix and match different tools from different frameworks in Galaxy. And I also find it an excellent thing how we do the data handling in Galaxy and in particular in Phenomenal. So what I briefly mentioned is that you can download the data from Metabolites. Metabolites was introduced to you by Kiva yesterday. And that download then comes with very rich metadata in the ISATAB format. And that will also be introduced to you in more detail by Philip Roccasera. It basically means that we have annotated all our samples with how they were generated and what experimental design factors are associated with one sample. And that then becomes interesting when you want to do the statistical analysis. And if nothing else, then it would give you which samples are blue and which samples are red in the PCA plot, for example. So we see that you start from metabolites. Via metabolites, we can go into the Galaxy and then create these outputs. And what I've shown in the demo is how to do that through the web interface, but we also have ways to do that on the command line. So you can actually fire up a complete workflow on a Galaxy instance through the command line. Now, this is a topic for probably more the bioinformatics geeks and not for bench biologists, but that way you can run a single workflow across a large number of studies. And this is exactly what Christoph has done with the statistics workflow we have in Phenomenal. That will also be introduced to you by Etienne later. So we mentioned running on the cloud. Maybe not everybody knows how expensive that can be because we don't know yet. So let's do some calculations on the back of an envelope. We mentioned the 24,000 samples earlier. Let's just say each one takes 1 minute to process, and that would be 400 hours on a single CPU core. Or if you break that down, that's roughly 2 weeks. So if you do that on your laptop, it might take 2 weeks. If we now try the cloud computing, then we can rent a CPU core for, let's say, 4 cents and put in another terabyte of storage and rent that for, let's say, $16 in the cloud. But why only do 1 CPU core? It is the same $16 if you rent 400 cloud servers, and you would be done in 1 hour. Or it's still $16 if you rent 24,000 cloud servers, 1 for each sample, and you would be done in 1 minute. I have to admit that is a lie, because firing up those 24,000 nodes will take a while. So there is probably more overhead than necessary if you do 24,000 cloud servers. But being done in an hour, that is across lunchtime. And the bottom line is that this all comes at constant cost. That is the important bit. And it is usually much more cost effective than running your own 24,000 CPU core HPC server in the basement. And if you think I'm making this up right now, you're right, I did make that up right now, but it has been done before. So this is from an article, and as you see here from the link, it's from 2011. So that's 7 years ago. And those guys, they were renting out 30,000 CPU cores, more than 20 terabytes of RAM and 2 petabytes of disk. So 7 years ago, this was a monster CPU, monster compute cluster. Of course, it costs a bit. It's $1,200 per hour. And they were doing that for some big pharma company, and they were running a large screening study, and it took a couple of days. But if they had done that on the in-house cluster, it would have taken weeks and months to complete. So this shows you that cloud computing is feasible. I just realized that I will have to rerun the MSMS to MetFrag step. Let's see where we find that, the MSMS to MetFrag step with the correct inputs. So that is this one. Because that job is waiting for something else. So let's see. What you didn't see is that Christopher Rutke's sent in a brief note that I shall do that. So let's see if we can do that while the demo. I have to ensure you, if I had taken the Galaxy tour that I introduced at the very beginning, I wouldn't have pressed the wrong buttons and I wouldn't have lost my way and I wouldn't have to go forth and back between presentation and the live demo. Okay, so I mentioned the prices of the cloud, but there is more than one cloud out there. And I have to admit that cloud pricing nowadays is a fair bit of a jungle. How much you pay depends on how many CPUs and how many RAM you're renting. That's very obvious. It also depends on where the cloud is located. So Amazon is having compute centers in Northern Virginia, in Ireland, in somewhere Asia, and they do have different prices. On Amazon, Germany is among the more expensive compute centers. There's also different flavors where you can buy a standard machine, or you can buy one that is reserved, as we see here. Now there you sort of have some kind of prepaid scheme for paying. There's other instances from some of the providers that are spot instances, and they are preemptible. That means if the cloud provider needs more compute power, they simply kill your virtual machine without any notice and give it to somebody else. Now that sounds like a nightmare, and we are not supporting that right now in Phenomenal. But if you have a big compute job, you see that the difference between the standard and this preemptible one can be fairly dramatic. And if your workflow can reschedule something that has been killed, then it is resilient to this killing of that instance, and it can simply restart the job somewhere else. And if we are talking about hundreds of cloud computers here, that really makes a difference. So next to the big ones like Amazon, Google, and Microsoft Azure, there's also more local cloud providers. For example, City Cloud is located somewhere in Sweden. They charge you 15 euro cent, which is on par with what you see here. There's also scientific clouds. I mentioned the Denby Cloud before. There you need to apply for a project, and it'll not cost currently, and it's part of the German Bioinformatics infrastructures. On the European level, there's the European Grid Initiative. And there again, you have to contact the people. They have different cloud providers in the backend, scientific clouds and others. And there the costing depends and can be anything between for free and the ones we have shown up there. So the summary, there is computational metabolomics tools to speed up metabolomics research. In Phenomenal, we have developed the cloud-based scalable software infrastructure. The workflow systems like the Galaxy I've shown help to crunch through the data analysis steps. And finally, cloud computing is feasible today. I didn't mention you don't always have to go to the cloud providers directly, but there's also consultants and resellers. And very often they have good relationships with universities and can help you optimize your cloud computing. Okay, with that, I have to do the acknowledgements, and especially the XCMS, MetFrag, and OpenMS workflow was quite a fascinating work to set up and implement. It's been done by Payam Imami in the group of Kim Kultima at Uppsala Hospital, and also Christoph Rübkes in my group. Now let's see if we can go back. Excellent. We now have the MetFrag parameters to run the MetFrag. Now we can execute that, and we now see we can really select those MetFrag parameters. I can execute that. We get some results, and then as part of the last step, I would then run the visualization. But maybe we can already have a look. James, what do we do? Do we have a query and answer now, or do we have the quiz now?
Hi, Stefan. We can do some question and answer. We've had a couple of questions. The quiz we'll hand out afterwards. So okay. It's up to you. If you want to finish your demo, we can do that.
I'm basically done. You see I have the Slack open either that way, or if people give their questions through the GoToWebinar. Which way do we prefer?
Either, it doesn't matter. We've had a few in the chat. People have been helping you out and answering them for you, but maybe you'd like to answer them as well. So the first question was, are these analyses made using LCMS or LCMSMS? This was right at the start of the talk. So, obviously, MetFrag uses LCMSMS, but I don't know if you want to add anything.
I can probably mention the way this has been done. This was using auto MSMS. So in auto MSMS, what the instrument is doing is it's acquiring a single MS1 scan, and then it's looking at the highest peaks in that MS1 scan, and then it's firing 1, 2, or 3 up to 10 data-dependent MSMS spectra that are targeting the, so to speak, the loudest precursors they found in the MS1 scan. And that way the whole LCMS run proceeds. So inside the LCMS file, you have both the MS1 and the MSMS information. We can also bring in separate measurements. So you can get the MS1 information from a pure metabolite profiling run. And then you might have dedicated MSMS runs maybe on your QC sample because you have more sample amounts that might be convenient. And some people are already trying and looking at SWOS, for example, which is some way to get not only 1, 2, 3 precursor peaks after an MS1 scan, but really all of the features are fragmented all the time. This is a bit more challenging, and we don't support that right now.
Okay, so obviously MetFrag uses LCMSMS, but for the LCMS and camera steps, can you do that with LCMS data?
Yeah, that's plain LCMS and requires on the MS1 information only. So you could run that workflow on LCMS data and simply skip or not do the MSMS parts. And then this will actually feed towards the statistics workflows that we're going to see later.
Great. And another question we've had is, can you state again, where is your CRE?
My what?
CRE, Cloud Research.
Oh, the one I deployed, it is not public, so you can go to that IP address, but you won't have a user account. If people want, I happen to be the administrator of that, and I've just pasted the URL into the chat window. You could then ask me for an account there, but in fact, it's not much different to the public instance that we are running. So you can simply also do that on the public instance of Phenomena.
Okay, we have another question. So why do you focus on this cloud computing setup when there are publicly available Galaxy instances, such as use galaxy.org or galaxy.e?
Mm-hmm. So one scenario is that you might be a hospital, and in the consortium we have the Uppsala Hospital, and we also have Imperial College with a medical facility. And when you're then talking about human patient data, then things become very sensitive. And I'm pretty certain that your data protection officer at Imperial College would do very nasty things to you if you uploaded patient data to the Galaxy server in the United States. This is why you can fire up your own Galaxy that is dedicated to you, and nobody else has access unless you provide an account. And you can even run that on the OpenStack cluster in the basement of the hospital. That's the reason why we focus on doing that on the cloud.
Could you also mention the fact that all the metabolomics tools are already kind of set up for you?
Yeah.
As opposed to if you use the galaxy.org, then you're not going to have XCMS and MetFrag ready.
Right, and I think that's something on the road for the next month and years, that we also need to get some of the tools into the public galaxies. Now, the workflow for Metabolomics team in France, they also have done an excellent job, and they already have a public galaxy that has many of these tools already installed. And that would be another fantastic place if you have non-sensitive data. I'm coming from a plant institute. Our plants don't have any privacy, so we can happily upload to W4M, for example.
Great, so there's been no more questions, so feel free to ask any further questions in either the Slack channel or in the chat for the rest of the day. We've just pasted in a link to a Google Doc with a multiple choice quiz where you can test your knowledge based on the talk today. So Stefan, if you got anything further to add, or are you done?
I probably have 2 more minutes because I'm now finishing the visualization part of the MetFrag workflow. So I selected the parameter files, which include the precursor information in the result file, which includes the ranked candidates. And this will now create the PDF for the results, and then we can have a look at that. No more questions in the chat. There is something that there's a link to the Google Doc in yours.
Yeah, so the Google Doc link in the chat window is a quiz, so have a go at that. Any questions or post them in the Slack.
In fact, what I can do is I can do some cheating. I was running the workflow yesterday, and you see, because I wasn't distracted by talking at the same time, I got everything right on the first go, and we can now visualize the results, and that'll now give us for all the individual precursors what they look like. There should be the structure, and what's biting me right now is that I have indeed been running a development version of Galaxy, and I didn't check the PDF yesterday evening. So that would be a bug that we need to fix, so that you can see the structure of the lipid here on the left in this PDF. We can also have a look at the Kubernetes cluster. So here you see how much CPU and other stuff was used in the last minutes, and you see everything was normal and fairly nice. Any more questions? If not, excellent. Thank you very much. Bye.